Hey guys, welcome back to LabMate. Today we're going to look into a medium that is important to urine analysis, which is CLED Agar, also known as Cystine-Lactose-Electrolyte-Deficient Agar. So whether you're a student, a lab tech, this is going to be a quick overview on CLED Agar. So sit back and let's get started. Okay guys, let's get started. So CLED Agar is also known as Cystine-Lactose-Electrolyte-Deficient Agar, and it is a non-selective and differential medium. This agar is mostly used in culturing urine samples or urine analysis. And because of the components within this agar, the colony morphology of the bacteria that's going to grow on this medium is going to help us in terms of narrowing down our investigations to figure out what bacteria has grown on it. So, simplify everything. We'll say that CLED Agar supports the growth of urinary pathogens because of the nutrients that are going to be in the agar itself. It stops Proteus species from swarming because of the lack of electrolytes, and it differentiates bacteria due to the pH indicator that is within the agar. Something very important to note is in a situation where maybe CLED Agar is unavailable due to a shortage, you can use McConkey Agar, preferably without salt, because McConkey with salt is going to inhibit the growth of gram-positive pathogens. And when it comes to CLED Agar does not inhibit the growth of gram-positives, but there are some gram-positive bacteria that might not grow, or how can I say this? They'll grow either slowly or they'll just be not grow at all, but all urinary pathogens should be able to grow. Let me continue on. So the components of this agar is lactose, beef extract, enzymatic digestive gelatin and casein, L-cysteine that leads to the growth of dwarf colonies called coliforms, bromomethyl blue, which is the indicator, and of course, agar. Quality control. So of course, after you prepare the agar, you will have a control plate, which is an uninoculated plate that is incubated to check for sterility. This control plate, I've basically mentioned it in all the previous quick overviews, and consistently it's going to be something you're going to need to do if you're going to be preparing your own medium to make sure that indeed it is sterile, and that when you perform any tests and there is growth, you know for sure it is coming from the patient's sample, not from contamination during preparation of the medium itself. So quality check organism is a lactose fermenting E. coli, because there are different strains of E. coli. So this one you will expect to see yellow colonies, Proteus mirabilis, to see that there's restricted swarming, and or Staphylococcus aureus, which is a gram-positive bacterium that should grow on this agar. Because like I previously said, it does not inhibit the growth of gram-positives, but some may have difficulty growing on it. Now going to uses and limitations. So of course, as I've been mentioning before, CLED Agar is used for routine urine culture, for helping diagnose patient infections. To differentiate between lactose fermenters and non-lactose fermenters, this is by using first the lactose that's within the agar itself, plus the bromomethyl blue indicator will help us identify the lactose and non-lactose fermenters. And also, CLED Agar is used to quantify and isolate microorganisms. So a lot of times when it comes to urine analysis, you'll see when a plate is being read, it will be quantification does occur, unlike with other types of culture where you just be looking at the morphology, like how does it grow? Is it a coliform? What is it? Right? So limitations of CLED Agar is that Shigella species may not grow on this medium, and the poor growth of some gram-positive bacteria, such as I mentioned before, such as group B streptococci. So these may need a blood agar to grow. So let's say, for instance, you do your analysis and you inoculate on both a urine plate and a CLED Agar plate. You might not see any growth on the CLED Agar because it might not grow. B streptococci might not grow well, but you may see growth on a blood agar plate. Also, fastidious organisms such as chlamydia and Neisseria gonorrhoeae will not grow on CLED Agar. So when it comes to result interpretation is because of the components of this agar, and every agar has its unique components, and that of course will affect how a bacteria may or may not grow on the agar. So when it comes to bromomethyl blue, when we see yellow colonies, we know that there were acidic conditions that occurred due to lactose fermentation. So for example, some strains of E. coli are lactose fermenters, right? So because not even some, majority are lactose fermenters. So these strains of E. coli, because they can ferment lactose, they're gonna byproduct, which is gonna be acidic, is gonna result in the indicator, which is bromomethyl blue, to shift from blue to yellow. And that's how you'll see, like, okay, that's what you'll see. But like I said, don't forget that some strains of certain pathogenic types of E. coli, such as O157, may not ferment lactose and will not produce a color change, so they will stay blue. So just like in an earlier video clip in the beginning of this video, you will see that it says non-lactose fermenting E. coli, and you see that it is blue. Meanwhile, in this photo on the side, this is an E. coli strain that can ferment lactose, thus resulting in yellow colonies. So that's very important knowledge to remember. Also, L-cysteine supports the growth of cysteine-dependent dwarf coliform colonies. That's also something significant to remember. And blue colonies can also be caused by bacteria that decarboxylate L-cysteine that cause an alkaline reaction. So instead, they will just stay blue because of the indicator. So, for example, Proteus mirabilis produces blue colonies. But sometimes we will see red agar or colonies because some bacteria may produce red agar or colonies due to increased alkalinity or peptone degradation. So, for example, Salmonella species may produce red colonies on CLED Agar. So, to summarize it all up, CLED Agar is a crucial medium in terms of urine analysis. It supports the growth of most urinary pathogens and helps us when it comes to identifying the bacteria. It's an essential tool in routine diagnostic labs despite certain limitations. It also helps us when it comes to quantification of the bacteria itself. So, while it's not perfect and has its limitations, the advantages it offers in routine diagnostic work makes it like a staple in microbiology labs worldwide. If you found this video helpful, don't forget to like, subscribe, hit the bell icon for more in-depth microbiology content. Thanks for watching, and I'll see you in the next video. If you want to see the source for some of our sources, you guys can check out the next slide where I do have my references, and I do also include some of them within the description of the video. Alright guys, bye!